Hello, everyone. Good afternoon. I'm glad to be here to present our work on activating transcription factor 3 as a potential molecular marker for spinal cord injury. And it also has a neuroprotective function. So let's see. All right. So I'm going to give a brief introduction of a spinal cord injury, and then the rationale we perform our experiment. And then I'm going to show you some data that hopefully will convince you that ATF3 is a molecular marker for injured neuron in spinal cord injury. And also I'm going to show the data that suggests that ATF3 has a neuroprotective function. And then I'm going to make a conclusion. So spinal cord injury, maybe it's not familiar with every one of you. It leads to a significant functional disability, including permanent motor and sensory deficit and autonomic dysfunctions. So there are currently 294,000 spinal cord injury patients now in the US, with 13,000 new patients every year. And the majority of more than 50% of the spinal cord injury occur at cervical level, and about 30% at lumbar level, at thoracic level, and 20% at lumbar level. The images here are the radiographic image of C6 fracture. This is the X-ray, this is the CT scan. And from the MRI, you can see very clearly there's a fracture, and then you see there's a swelling of the tissue being compressed by the bone. So what happened after spinal cord is injured? So there's usually a primary injury caused by mechanical insult, followed by secondary injury, which is featured by neuronal injury and cell death, and demyelination, axonal degeneration, and neuroinflammation. And neuronal injury is actually one of the most important neuropathology after spinal cord injury. So, and currently there's still no effective therapies, and most of the clinical trials failed to find effective treatment for spinal cord injury. And partly because we still have a poor understanding of the mechanism of neuronal injury early after spinal cord injury. And therefore, it is urgent to find a novel marker that allow us to label the injured neurons after SCI, and that allow us to target the early response to injury to save those injured neurons from dying, and therefore promote neuroprotective and regenerative response in the injured tissue. So we started the project with a broad question: what is the early transcriptional response to spinal cord injury? So we use RNA sequencing method to try to profile the gene expression at 4 hours after injury. We choose 4 hours is because at this time point, the injured neuron can still be rescued. So we use a mouse model of a unilateral cervical C5 contusion injury. We have sham versus injury at 4 hours using wild-type mice. And we prepared the total cellular RNA, and we submitted to the functional core facility at UCSF for RNA sequencing. So we got about 30,000 genes and transcript from the assay. And then we further filtered the genes and retain those are differentially expressed. So the total list number of genes with the differential gene expression with 1.5-fold changes, either upregulate or downregulate, is about 2,000 genes. And then we further filtered with multiple correction, Benjamin-Hochberg false discovery rate. That is adjusted p-value. So then we obtain about 177 genes, and 160 genes are upregulated and 17 genes are downregulated. So then we did a heat map. This is a heat map of the differentially expressed genes with multiple corrections. So as you can see, it's about 10% of these genes are downregulated after injury. And the majority of the genes, 90% of the genes, are actually upregulated after injury with 3 replicate in each condition. And I just selected the top genes with high fold changes from that list. And then ATF3 is among one of them. This is a volcano plot of the differentially expressed genes among the 177 genes. And as you can see, ATF3 is one of the most upregulated genes with about 10-fold increase. And with quantitative RT-PCR, we validate the RNA-seq result. The fold change is actually a 25-fold increase by RT-PCR. So what is the function of ATF3? So ATF3 expression in normal tissue is low, and it is an early response gene that induced after injury or stress. And as a matter of fact, ATF3 has been used as a marker for injured neurons in peripheral nerve injury. And actually, one of the studies in the literature have shown that following sciatic nerve injury, ATF3 is pro-regenerative. You can see they use a transgenic mice which overexpress ATF3. And then you can see these nerves after injury in the transgenic mice that overexpress ATF3, they have more robust nerve regrowth than the wild type control. And that is the peripheral nerve injury. And then in central nervous system disease, for example, ALS, ATF3 has been shown to improve motor neuron survival. As you can see, there are 2 types of transgenic mice, and then the G93A. This mice, before 60 days, this mice has normal number of motor neurons. And then after, at 90 days to 120 days, you see these motor neurons, which is the green color, they died. Whereas if the mouse has also overexpressed ATF3 genes, and you can see that at 90 days and 120 days, these motor neurons still survive. So which suggests that the presence of ATF3 can save motor neuron from degeneration. And also in other model like TBI, they show that ATF3, if you delete ATF3 genes, you see more bleeding, hemorrhage in the knockout mice compared to the wild type mice. And also the mice show more inflammatory response. So, however, the function of ATF3 in spinal cord injury model is still largely unknown. So we hypothesize that ATF3 promote functional recovery and can reduce tissue damage after SCI. So we did a couple of behavioral tests paw placement and grid walk, as well as histology. We measured the injury area and doing immunohistochemistry to label the cells that express ATF3, as well as biochemical assays like Western and qRT-PCR. And this is our finding. So we use a global ATF3 knockout mice. And then, so we did, the first test we did is the paw placement, contralateral paw placement. So basically we put the injured mice at different time point after injury into a cylinder. So the mice will stand up and use their arm to, their forelimb to support them on the cylinder. And then when we measure the frequency, they use the uninjured side. So the more they use the uninjured side, the contralateral side, the worse the injury, right? So they use the injured side less. So you can see that ATF3 knockout mice use more of the healthy side, so which suggests that the recovery is worse. And then we also measured the foot drop during the grid walk. So the more they have foot drop, suggests that they have a worse recovery. So ATF3 knockout mice has more foot drop than the wild type controls over 2 weeks. And we also take the cross section of the injured cord, and we look at the sections with the maximum injury area. And you can see that ATF3 knockout mice has a significantly enlarged injury area than the wild type controls. So next question is, where are ATF3 expressed? So we mainly look at the expression of ATF3 in neurons, which is neuron positive cells. And we also look at microglia, macrophages, astrocyte, and oligodendrocyte. Those are the glial cells in the central nervous system, which I didn't include the data here. So basically, I'm going to show you the neuron staining. So this is a immunohistochemical staining of the hemichord. So you can see this is actually half of the cord, hemichord. And then the green fluorescent signals label NeuN, which represent the neuron. And the red channel is ATF3 staining, okay? And this is low power, and this is the higher power. And as you can see, all the ATF3 positive cells, which is the red dots here, are NeuN positive. They co-localize very nicely. So ATF3 looks like it's only expressed in NeuN positive neurons 1 day after spinal cord injury. And then the next question is, what is the potential mechanism involved in the ATF3 mediated neuroprotective effect? We looked at several regeneration associated genes, including SPR1A, the small proline-rich repeat protein 1A. And it is one of the group epithelial differentiation genes. SPR1 expression is low in uninjured neurons, but is induced after injury, like exotomy. And it has been shown that ATF3 can promote nerve regeneration. If they use actually a SPR1A knockout mice, and then they did a sciatic nerve injury, and they found that reduction of ATF3 function actually limit axonal outgrowth. So then we test our sample, and we found that by RT-PCR, we found that the expression of SPR1A in uninjured condition are pretty low. And then upon injury, in both wild type and knockout mice, they are significant increased. However, the increase is actually significantly reduced in the knockout mice compared to the wild type mice. And we confirm the PCR result with Western blot. And you can see the protein expression of SPR1A is low in control, and they are increased after injury. However, the increase is reduced because of ATF3 deletion. So this suggests that SPR1A is ATF3 dependent. Its expression is ATF3 dependent, and it may be associated with the ATF3 mediated protective effect. And then we actually also did this in using an ischemic stroke model by our collaborator. And so here is the histology. You can see that the cross-section of the infarct area, well, it's the whole brain, but look at the infarct area, you can see that the infarct volume here in the knockout mice are significantly larger than the wild type mice. So in summary, ATF3 expression in sham control animals is low, and it is quickly induced after spinal cord injury. Early after injury, for example, 1 day post SCI, ATF3 expression is only induced in injured neurons, and therefore it has a potential to be a molecular and cellular marker for neuronal injury. Using the global ATF3 knockout mice, we show that ATF3, our data suggests actually ATF3 can promote neurological function and reduce tissue damage. So in the future, so we would like to explore, look at the neurons, the transcriptional response in specifically in neuron by using single cell sequencing technique. And then we can hopefully we can tease out the genetic target that's specific to ATF3 effect, and therefore find those targets for potential neuroprotective and regenerative strategies and intervention. Last but not the least, I would like to thank my collaborators in Brain and Spinal Injury Center at the UCSF. And then my collaborators in my department, Dr. Guang's lab and the Center for Cerebral Vascular Research, and also the colleague and scholars in my lab as well, and our funding agent from FAIR and UCSF Chancellor's Funds.